Hello everyone, my name is Jaziba Bahri and my presentation is on the genetics of circadian rhythm coordinating plants' response to environmental stressors. I was interested in the topic of circadian rhythm because I remember it being brought up several times in my previous biology lecture classes, and it seemed to be a very important topic because it dictates what biological processes happen in an organism and at what times, basically setting an internal schedule so that the organism functions properly in relation to its environment. I wanted to focus on circadian rhythm specifically in plants because even though we know that they're alive, it can seem rather strange to think of them as operating on some sort of internal schedule when they don't have a brain or can't move the way that we do. Finally, I wanted to examine what happens if the circadian rhythm in plants is disrupted or otherwise stressed. So this is an outline as to what topics I'll be covering in my presentation today. First, I'll start with what circadian rhythm actually is and how plants fit into the history of chronobiology, which is the field of research or study in biological timing, such as circadian rhythm. Then we'll go into what processes in plants are affected by circadian rhythm and how it is governed in plants on a cellular level and what important clock genes are involved. In this, we'll be looking at how this is operating in Arabidopsis thaliana in particular, a plant species that is considered a model organism because what we learn about the circadian rhythm in Arabidopsis can be applied to how we study the circadian rhythm in other plant species. And we'll be examining this further through our experimental study, where we'll be looking at what we know in Arabidopsis and circadian rhythm in that, and how we study the circadian rhythm in soybeans and how that affects their response to drought. Finally, we'll wrap up with a take-home message. So first we're going to discuss what circadian rhythm is and exactly how it comes about. So when the Earth rotates around the sun, this causes differences in the amount of light hitting a given location on Earth at different times of day, and this also affects temperature fluctuations in that given location throughout the day as well. So this generates our day-night cycle, and organisms have to be able to respond to these daily environmental changes. This is where the circadian clock comes into play, or the circadian rhythm, as it acts as our internal timing device to be prepared to make these adaptations in preparation for these daily environmental changes. So circadian rhythm operates on an approximately 24-hour cycle, and this is indicated by the name itself as it is derived from the Latin phrase circadian, which means about a day. It is present in virtually all species, so most species have circadian rhythm, and circadian rhythm also is driven at a cellular level by specific genes which are known as clock genes. The circadian rhythm is prompted by environmental cues, and these cues are known as Zeitgeber, which in German means time giver, and these are simply external cues that set or synchronize the circadian clock. Typically, this is going to be the day-night cycle, but it can be other external factors such as interactions or temperature, which can all impact a cycle's phase. The field of research related to the study of biological timings, such as circadian rhythm, is known as chronobiology, and we'll be going into that on the next slide and how plants relate to it. So chronobiology only really became a proper field of study about 50 years ago, but people have been interested in studying the biological rhythms or biological timings of different processes within organisms well before that, including studying the biological timings of plants. So one example is the French scientist de Mairan, who lived in the 1700s and was studying the daily raising and lowering of leaves in one plant, which is speculated to be Mimosa pudica, otherwise known as shame plant, shown on the right here. It has this fun behavior where if you touch its leaves, the leaves will fold in on themselves, but de Mairan was specifically focused on how this plant raised and lowered its leaves throughout the day. And de Mairan found that even when the plant was placed in a dark room, so away from the sunlight and away from any other potential environmental changes, that even under these constant conditions, it was still raising and lowering its leaves throughout the day at specific times. So this is indicating that this behavior is being driven by an internal clock that works under constant conditions rather than just be strictly a response to the sun. So the timing of many different processes in plants are affected by circadian rhythm. This includes photosynthesis, plant leaf movement, as shown previously in Mimosa pudica, as well as in other plant species such as prayer plants, which is shown here. Circadian rhythm can also impact growth, as well as how the plant responds to different environmental stressors, such as chilling stress or frost or water deficit from drought. Flowering can also be affected by circadian rhythm, as it can impact how many flowers are blooming, as well as the timing of flowering. So what we currently understand about how circadian rhythm is governed in plants at a cellular level is through a study of the circadian clock in a plant species known as Arabidopsis thaliana, otherwise known as thalecress. And Arabidopsis is considered a model organism because what we understand about the circadian clock in the species can be applied to how we study the circadian clock in other plant species. However, even just how we modeled the circadian clock in Arabidopsis has undergone a lot of changes in development over time. Because when we first were studying the circadian clock in Arabidopsis, we had set the general model to be set in a linear flow consisting of 3 different modules. The first one being the inputs, such as the cues from the light-dark cycle or in temperature, that would cause input pathways that reset the central oscillator. The central oscillator would then generate the circadian rhythms and cause for different outputs to be put into place, where they would control the timings of different biological processes as shown on the right here, many of which I covered previously in the last slide. However, this process isn't as linear as once thought, since these modules can actually feed back into the previous one and alter it to some degree. For example, the outputs here feed back into the oscillator to modulate the central oscillator's function, and the central oscillator itself can sort of pick and choose how to interpret or when to interpret these input signals. And this is known as a gating mechanism because the clock can be made to be more sensitive to certain inputs during specific times of day, which allows the clock to vary the level of intensity of the plant's general response to these different inputs depending on the time of day. So the clock genes that run the circadian clock in Arabidopsis thaliana have also been identified over the years, and these are some of the most important ones that make up 3 different loops that all feed into one another and all work together to make sure that the circadian clock runs properly. These genes will operate at different times of day and impact one another to regulate their level of expression. So first we have our morning genes, which consist of CCAI1, which stands for circadian clock associated 1, and LHY, which stands for a late elongated hypogonadal. So these are considered to be the morning genes because their peak transcription level occurs in the early morning around dawn, and so they're copying information from our DNA to RNA in the process of making proteins. And these genes will also regulate the level of expression expressed by TOC1 or PRR1, which makes up the evening genes. So TOC1 stands for timing of CAB expression 1, and this is considered to be a part of the PRR family, which is a group of genes that stand for pseudo-response regulators. These genes are considered to be evening genes because they peak in the early evening. One interesting fun fact about TOC1 is that a study has shown that TOC1 impacts the speed of the cell cycle in Arabidopsis. And since it impacts the cell cycle, this impacts the level of growth and development experienced by Arabidopsis, depending on how much TOC1 is expressed. So when TOC1 is expressed normally, then growth and development will happen normally. But if there's an overexpression of TOC1, then the cell cycle actually slows down, which limits the amount of growth because the cell cycle isn't happening as quickly, and so cells aren't dividing as quickly. Additionally, conversely, when TOC1 is underexpressed or has otherwise lost its function, then the rate of the cell cycle will increase rapidly, and this promotes faster growth in Arabidopsis. So CCAI, LHY, and TOC1 all form the central feedback loop of the central oscillator, and these morning and evening genes were first found and included in the circadian clock model in 2001. The evening complex was found a little bit later much later in 2011, and was then included into the circadian clock model that was shown previously. And these genes are expressed from the evening throughout the night and consist of LUX, which stands for Lux Arrhythmo, ELF3 and ELF4, which stands for early flowering 3 and 4. And these genes are primarily responsible for regulating the expression of these early morning genes shown here. So as you can see, these genes in the different loops will regulate the expressions of the other genes, which is necessary for the circadian clock to operate properly and work accurately in its response to different environmental factors. So it is all well and good that we can model the circadian clock in Arabidopsis and understand the clock genes involved. But if it's a model organism, then what we know about the circadian clock in Arabidopsis should be applicable to our study of circadian clocks in other plant species. So this brings us to our experimental study, which is based on the 2013 paper by Marcelino Gomez et al. And their study titled Diurnal Oscillations of Soybean Circadian Clock and Drought-Responsive Genes. So what this title is referring to is that they were studying the soybean circadian clock and drought-responsive genes and their level of expression throughout the day. So first we have to go into a bit of background as to how plants deal with drought. It's important because water deficit induced by drought results in many molecular and physiological changes in plants so that they can better protect themselves from the repercussions of it. Going back to our model organism, Arabidopsis, their drought response mechanism has also been well studied. In Arabidopsis, this mechanism is split between two different pathways, one that is abscisic acid or ABA dependent and one that is ABA independent. Abscisic acid is a plant hormone that is very involved in growth and how Arabidopsis and how plants in general deal with environmental stressors. And I've included a molecule of one here. And between the two different pathways, they involve different transcription factors, but both have these transcription factors bind to specific elements that induce stress-responsive genes, which would then encode different protein classes to ensure that the plant survives this stress. One example is galactanol synthase, which is an enzyme involved in the biosynthesis of raffinose, which helps the plant survive extreme osmotic stress. So the researchers were specifically interested in how drought affects crops, particularly soybeans, because soybeans' genetic and molecular behaviors have been well studied, but its circadian oscillator and just generally how its circadian clock works has not. And this is important because drought can act as an environmental cue that can modulate the circadian clock, which in turn impacts the synchronization and timing of different molecular processes within soybean. In turn, this would impact the timing of flowering and how many flowers are being produced within a given soybean, which would then impact the amount of yield of seeds, and this would be of importance to farmers who are trying to make a profit. So the researchers did not have an outright stated hypothesis, but they generally thought that drought would act as a sort of environmental cue that would impact the circadian clock and how it would affect the timing of these drought response genes. So to conduct their experiment, the researchers inflicted 2 different treatments on the soybeans. First, a drought treatment, and second, an ABA or abscisic acid treatment. For their drought treatment, they first germinated the soybean seeds by having them grow in these peat pots within a growth chamber for 15 days under normal growing conditions. And this consisted of spending 14 hours in the light at 28 degrees Celsius and then 10 hours at night at 20 degrees Celsius. And this was done to mimic the day-night cycle. The seeds would also get a regular amount of water so that they would grow normally until they reach the V2 stage, which is shown here, where their leaves have sprouted. And at this point, they would be separated into 3 different groups and get different water treatments. So they calculated the level of soil moisture under these different treatments through gravimetric humidity, which is abbreviated as GH, and this is simply the percent of water present in the soil in relation to the dry weight of the soil. So for example, in the unstressed treatment or the control, this had a 70% GH, so this means that there was a good amount of moisture in the soil and the percentage of water within the soil was relatively high. Compared to that, we have the moderate stress treatment, which has a 30% GH, so the soil is a bit drier, is much drier compared to the control. And then you have the severe stress treatment, which is extremely dry at 15% GH. So after this, they would let these soybeans grow under these conditions until they reach the V1 stage, upon which they would collect the leaves at different Zeitgeber times and 4-hour intervals throughout the day, collect them and then freeze them in their sampling so that later they would isolate the RNA and see which genes are transcribing at different Zeitgeber times. So this the next treatment that they had was the ABA treatment, and since ABA was important in the drought response mechanism in Arabidopsis, they wanted to see if this also applied for soybeans. So the beginning part of this experiment was similar to the drought treatment, where the soybean seeds would grow in peat pots in a growth chamber for 15 days under normal circumstances under the light-dark cycle at different temperatures until they reach the V2 stage. And then the abscisic acid treatments would be inflicted at Zeitgeber 3 specifically, and this is the same as 11 o'clock a.m. And the plants were split into 2 different groups, one that received a mock treatment, which is the control, and the second group that received the ABA treatment, where they would get sprayed with this mixture of 100 millimolality of abscisic acid dissolved in a mixture. Afterwards, they would collect their samples at different Zeitgeber times throughout the day and isolate their RNA transcripts again and see what genes are being actively expressed at different times. It is important to note that the researchers also had to find the circadian clock genes in soybeans by identifying their homologs to the Arabidopsis circadian clock genes via an online tool called Blast Search Tool. And what I mean by homologs or homologous genes is that these same circadian clock genes present in or Arabidopsis should also be found in soybean with the same origin and the same function, even if they have a different name. So the researchers found that there were 11 homologous genes identified for the different circadian clock genes. So this slide shows their results from finding how the different circadian clock genes in soybeans reacted in relation to the moderate drought stress and the severe drought stress. So since they identified 11 of them, we're only going to look at a couple of them, specifically the ones that the researchers took special note of. So first we are going to look at the results of the expression by GMTOC1 and GMLUX. And if these names sound familiar, it's because these are the circadian clock genes in soybeans that are homologous to the TOC1 and LUX genes in Arabidopsis. So if we look here on these graphs, these are on the x-axis, we have our Zeitgeber times, starting from 0, which is when the lights first came on, up until 24, which was the end of the cycle. And our y-axis, we have the level of expression of these genes throughout different times of day. On the left side, these are the genes under the moderate drought stress. On the right, we have them under the severe drought stress. And the bolded lines indicate the control plants, and the dotted lines indicate the stressed plants, the plants affected by the drought stress. And so if we look on the left side where we're comparing the control plants to the plants affected by the moderate drought stress, there really isn't that much difference in level of expression throughout the day for GMTOC1 or GMLUX, since they seem to be fairly closely aligned in position. But if we look on the right, something very different has happened in the severe drought stress, where if we look here, these peaks of expression in both GMTOC1 and GMLUX have become much smaller and have peaked earlier in the day rather than at their normal time. And this indicates that this advanced peak expression in the evening genes has also happened in previously studied Arabidopsis mutants that didn't have CCA1 or LHY. And if we remember how the clock genes work in Arabidopsis, the CCA1 and the LHY, these morning genes, are regulating the level of expression in TOC1. So if this level of expression in soybeans, the GMTOC1 or the GMLUX, they are likely affected by the level expression of the morning genes in soybeans as well. So if we look there, we find that our gene that is equivalent or homologous to CCA1 or LHY, the morning genes in Arabidopsis, the equivalent gene in soybeans is GMLCL1. And if we look here as well, their level of expression has also been heavily affected by the drought stress. Interestingly, if we look on the right side first, under the severe drought stress, the level of expression for the plants experience the drought stress is much lower in the time that GMLCL1 is supposed to be active if it's supposed to be a morning gene, since it's supposed to be active during this time, in comparison to the control, which has a much higher level of expression. But when we look to the left side where the moderate drought stress is present, the plants under the drought stress actually exhibit higher levels of expression compared to the control. And the researchers believe that this slightly higher level of expression under moderate stress at earlier times could be due to this gene potentially being involved in the signaling and activating drought response mechanisms at an earlier time in the day. So if we look at the drought response genes in soybeans on the next slide, we'll see that in both the moderate drought stress and then under the severe drought stress, these genes their level of expression is oscillating throughout the day. So they're not being expressed at the same levels all the time, they're changing. But even so, a lot of their they exhibit higher level of expression in both the moderate drought stress and the severe drought stress earlier in the day, which may coincide with the higher level of expression of GMLCLI. So next we have our results from our abscisic acid treatment or ABA treatment. And if we recall from before, abscisic acid is an important hormone in Arabidopsis in mediating one of their pathways for drought response. And the researchers are interested in seeing if abscisic acid is also important in drought response for soybeans. And so they found that only a couple of circadian clock genes and most drought responsive genes were affected by the abscisic acid treatment, which is indicated in the dotted lines and can be compared to the control plants in the solid black lines. At least for the drought responsive genes, they seem to have been affected, and particularly GMRAB18, which gets this peak for the abscisic acid treated plants earlier on in the day. Meanwhile, the circadian clock genes weren't too much affected apart from GMTOC1 and GMJumanji, and the researchers noted GMTOC1 in particular interest because at Zeitgeber 16, the level of expression by GMTOC1 under the abscisic acid treatment is lower than the control. And in Arabidopsis, at least, the homologous gene TOC1 is part of a feedback loop that modulates abscisic acid responsiveness. So the fact that this is lower, they believe, warrants further study. So the researchers confirmed through their study that drought stress does indeed affect the gene expression of the circadian clock genes in soybeans, and they found that the circadian clock genes may have a sort of potential regulatory interaction between them and the drought responsive genes. And this is because many of the drought responsive genes involved in plant defense against the drought stress exhibited higher expression during or before dawn, which corresponds to the higher level of expression of morning genes, at least under the moderate drought stress. And this may be a result of an interaction between the cell dehydration defense genes and the circadian clock genes to optimize the plant metabolism to earlier times in the day, which would increase the plant survival. And this study is important because, once again, the productivity of crop species is directly linked to their flowering capacity. So this is how much they flower, and that would impact the amount of seeds that would be produced. If we can understand how these interactions work, then perhaps through genetic engineering, we can further manipulate the circadian clock and improve drought tolerance and thus improve productivity. So I know a lot of information has just been covered, but the main things that I would like you to take away from this is that circadian rhythm is the internal clock that synchronizes an organism's biological processes, that Arabidopsis thaliana is an excellent model organism since its circadian rhythm model is applicable to other plant species, and that patterns of gene expression in Arabidopsis appear to be the same in commercially viable species like soybeans. So further research in plant circadian rhythm has great potential in improving crop yield. So thank you all for listening to my presentation, and I hope you have a wonderful day.